following objective is to understand the role of enzymes in chemical reactions. And it's really nice that we just talked about proteins because enzymes are an example of a type of protein. So all that we have said about the structure of proteins, the possibility of denaturing proteins if there's excess acidity or excess heat, those will hold true for enzymes and their structure and their function as well. So enzymes are essentially catalysts and they function to speed up the reactions in our biological systems. They basically make processes go faster and more efficient. They increase the rate of chemical reactions. So essentially, when we break food down, when we transport molecules around, when we convert one substance, one structure to the other, when we have cellular respiration, for example, which requires enzymes. When we think about the rates of those reactions, they would proceed much slower if enzymes were not present. So they function by decreasing the activation energy for a specific chemical reaction. When we say decrease the activation energy, they make it more favorable. They make it easier. They lower the barrier of how much energy is required to drive this reaction in the forward or even reverse direction. The way that enzymes do that is by binding to reactants. And reactants are substrates, but they can also be, enzymes can also bind to the products as well. We'll talk about that. So enzymes can allow a reaction to proceed in the forward direction, which is when we bind to the substrate and result with product, or enzymes can allow a reaction to proceed in the reverse direction as well. So if there's a lot of product around, the enzymes can bind to the product and that can catalyze the reaction, which converts that into our initial substrate. So what we want to really emphasize here is that enzymes, depending on the level of substrate and product, the reaction will proceed either in the forward or reverse direction. And this ties into what we discussed before with homeostasis. If we're getting to a scenario where there's excess substrate building up on the left of our equation, that is going to throw the homeostatic balance off. And so what happens is enzymes will bind to the excess substrate and shuttle that off into becoming or catalyze that into creating product on the other side of the equation. And the opposite of true. If there's excess product, the corrective action or the negative feedback action is to convert that rather into substrate. So what do we mean by saying the activation barrier is lower? So if we look at this graph, it's showing us the energy that's required for the reaction to proceed, and it's showing us the reaction coordinate. In other words, time. How does the reaction actually proceed? Now, if we compare what happens with the presence of an enzyme, this is the curve in blue, versus the absence of the enzyme, which is the curve in red. So we're looking at how much energy is required to go from reactants, if we take the example here, which is carbon dioxide and water, and we convert that to carbonic acid, which is the normal reaction that would occur with those two substrates. This is the product that we would get. Now, if there's no enzyme present, notice how much more energy. It actually takes twice as much energy for the reaction to proceed from our reactants or substrates to product. If the enzyme is present, the activation energy is lowered. It takes significantly less energy, half as much energy for the reaction to proceed. And then our products are formed on the right. So notice that the overall energy released during the reaction, right? And then notice the amount of energy that's required in the presence of the enzyme, which is the activation energy, versus the absence of the enzyme, which is twice as much energy that needs to be invested to drive this reaction. Let's talk about the way that substrates and enzymes can bind and interact and carry out these reactions. So there are two models for the way that enzymes bind substrates. And when we talk about models, these are basically theoretical concepts that were derived from, scientific experiments and so forth, but they're not actually proven. They haven't been visualized, so to speak. And so that they're basically models or theories for how this binding occurs. So the first model or theory is the lock and key model. Now, in the lock and key model, we basically have an enzyme which has a very unique fit for the substrate. So here's our enzyme. Here's the activation site on the enzyme, which is the spot for the substrate to bind. And notice that there's a unique fit to only one type of substrate or one type of molecule. So we call this the lock and key model, which is very similar to a lock on a door and a key being a very unique fit in order to open that door or that lock. So this theory explains the specific binding that we see, which we know is true. When we have an enzyme present in the body, it doesn't just bind to anything, right? It binds very specifically to the substrates that, or the reactions that it catalyzes. It will only bind to the substrates involved in those reactions. So that explains the lock and key specificity, but what it doesn't explain is the reversibility of most enzymatic reactions. If we're saying that the enzyme can catalyze the reaction in the forward direction and the reverse direction, the enzyme should be able to bind to the substrate as well as the products to catalyze that in the opposite direction. So the lock and key model explains some aspects of enzyme specificity, but it doesn't explain the reversibility that we see with enzyme reactions. The second model here or theory is the induced fit model. And this one is a little bit more plausible. It's a little bit more believable. It explains that the enzyme has the activation site, which is the spot for the substrate, and it can shapeshift. So the activation site can be modified to induce a fit to the substrate. So here's our enzyme, which has been modified. The activation site can now bind specifically to the substrate, and then the reaction can proceed and create the product on this end. And this will now explain the reversibility because our product, which looks differently than the initial substrate, can induce a fit. So when the product gets in close proximity to the enzyme, that will shape-shift the molecule or the enzyme, and that will allow for binding, and then the reaction can proceed in the reverse direction as well. So the two models explain certain aspects of enzyme binding. The issue with the product binding, or excuse me, the issue with the induced fit model or the criticism of this model is that in theory, if any substrate or product got in close enough proximity to the enzyme, it could induce a fit, which doesn't quite explain the specificity binding, right? So both of these theories have some, criticisms, or we could poke holes in some of these theories in different ways, but they're the most common and popular theories about how enzymes behave, and individually, they very well explain different aspects of enzyme binding. Let's look at cofactors and coenzymes. These are molecules that are needed in order to help enzymes function properly. Cofactors are going to be more directly involved, so they're non-protein components of enzymes, but they're necessary for the enzyme to hold its function, to hold its conformation, or some aspect of the way that the enzyme is going to behave. Examples of these are going to be trace minerals, magnesium, copper, zinc, or even iron. Now, when we think about these trace metals, these can essentially help catalyze or stabilize the enzyme for it to be able to behave or carry out the reaction. An example of this is DNA polymerase. So DNA polymerase needs magnesium as a part of its proper function. So in the absence of magnesium, DNA polymerase cannot, actually transcribe our DNA, and so it's going to malfunction. And so cofactors are going to be extremely important in the actual enzymatic process. Coenzymes, on the other end, are non-catalytic. they're not actually involved. They're kind of just playing a supporting role, right? They're kind of like behind the scenes in this, in their action. They just support the action of the enzyme, but they are needed for the proper enzyme functioning in an indirect way. They can be derived from vitamins. They can be smaller transfer molecules. A good example is NADH. We talk about NAD as one of our nucleotide units, and NAD can carry electrons around, and electrons can be required for proper enzyme functioning. So the way that I like to sort of separate these two is I like to think about the cofactors as integral components. So if you're driving a car, the steering wheel is a cofactor. You cannot drive the car without the steering wheel in place. The coenzyme would be something less essential, like maybe the headrest. The headrest is going to make you more comfortable as you're driving, but you can drive the car without the headrest. The headrest is really just supporting in the movement of the car. The steering wheel is required. It's an essential component that is actively involved. Let's look at the enzyme reaction rate. So the way that we can modify or regulate the speed of reactions. So these can be affected by several different factors. Enzymes catalytic rate. So the initial, not initial, but the innate catalytic rate of an enzyme. How many molecules can be generated per time? Another factor is going to be the substrate concentration. If we have lots of substrate, we're going to have a faster rate of that reaction current. The enzyme concentration, likewise, if we have a lot of enzyme, if they're saturated with enzymes, that is going to allow the reaction to proceed faster. The affinity of the enzyme for the substrate. So in order for enzymes to carry out reactions, there has to be a strong affinity or affiliation of the enzyme to substrate, and that can change depending on different factors. Temperature is one. That can make the enzyme less likely to want to bind to the substrate or product. And so the affinity is another factor that can speed up the rate of the reaction. Lastly, temperature and pH. We know that enzymes are denatured under acidic temperatures and not acidic, under higher temperatures and acidic pH. And so if the enzymes are in extremely high temperatures or extremely acidic pH, they can be denatured. However, just general higher temperatures creates more catalytic activity and can actually speed up the reaction to a point before denaturing the protein. So let me say that in another way. The temperature and pH can actually speed up the catalytic energy within the enzyme. However, if that gets beyond a certain threshold, that can actually denature the enzyme. Let's look at the way that we can regulate enzyme activity. Now, if we think about enzymes, they're always existing in our body. So why aren't they always catalyzing reactions? What is it that turns an enzyme on or turns it off? What are some of the factors that can regulate enzyme activity? So there are four factors that we're going to look at. There's the allosteric regulation, covalent regulation, which means binding, right? Covalent binding can turn that on and off. Feedback inhibition and feed-forward activation. These two are going to be essentially determined by the amount of substrate or product. So that can feed forward the reaction, or it can inhibit some of the earlier substrate or steps in the activity, in the reactions. Let's start with the allosteric regulation. And this is basically turning an enzyme on or off with the binding of a separate site. So there's the activation site, which is the site for the substrate or product to bind. And then there's a regulatory site on the enzyme, which means that some other modulating protein or modulating molecule can bind to the enzyme and can turn it on or turn it off. So here is the enzyme on the other end with the modulating structure bound, and that is going to have altered enzyme activity. As we can see, the activation site has now been modified. So essentially, other molecules can bind to our enzymes and turn them on or off, and this is allosteric regulation. Then there is covalent regulation, and this one involves another type of enzyme. So here's our enzyme in question, and this is the enzyme that's actually catalyzing the reaction. Here's the activation site. Here's a chemical group. So some type of chemical group can be added to the enzyme, but in order for that to be added, there's another enzyme that is required, which actually catalyzes the covalent bond between the chemical group and the enzyme in question. Essentially, enzyme A is needed to add this chemical group onto the enzyme that we're looking at. And when that happens, we now have the enzyme with an altered activity. It's in an altered state now that it has a covalent bond to the chemical group that is in question. In the reverse, the opposite occurs. So there's another enzyme, enzyme B, which is needed to take that chemical group off of the enzyme in question, and it's going to bring it back to its original state, which is not having that covalent bond to the chemical group. An example of this is using protein kinase and phosphates. So the enzyme that catalyzes the addition of the chemical group in this example will be protein kinase, and the chemical group in question here will be a phosphate group. So a lot of our enzymes, especially the ones that we'll look at when we talk about glycolysis, these enzymes will have the ability to add a phosphate group. And in their phosphorylated state, they're going to have a different state. They're going to have a different, they're essentially going to be turned on or activated when they have a phosphate added, and that is done by protein kinase. In the opposite direction, we can deactivate this enzyme by removing the phosphate group. And in order for that phosphate group to be removed, a phosphatase is required. So when you think about these terms, this is going to carry on when we look at other aspects of the course and other systems. We'll talk about several different enzymes that behave in this way. And so it's an important rule of thumb. When you hear the word kinase, it's going to add a phosphate. This is always true. And so when you talk about muscle contractions and so forth, you're going to talk about kinases. And they basically add phosphates and turn the enzymes on. Alternatively, when you hear the word phosphatase, that is going to cleave the phosphate off. It's going to cut that covalent bond, and it's going to remove the phosphate, and it's going to bring the enzyme back into its original state or turn the enzyme off. Next, we can think about feedback inhibition. This is an example of negative feedback. If we think about the reaction happening here, the rates of enzyme 1 and enzyme 3 are pretty much stable, right? These are not changing. The rates of enzyme 2, however, is dependent on how much of substrate C is present. If we have a lot of substrate C, that is going to deactivate enzyme 2. And it's essentially going to stop the reaction from proceeding because we're building up too much of the substrate C. And so the rate limiting step can be described as the conversion of substrate B into substrate C or product C, depending on how you look at it. So from B to C, this is the rate limiting step. And many reactions have a rate limiting step. It's the step that will ultimately determine the rate of the entire reaction. Therefore, if we have lower levels of substrate C, that can actually speed up the action of enzyme 2 in order to produce more of that substrate C. Here is another example of feedback inhibition. So this is a different reaction. Here we have the action of enzyme 2 and 3 being constant. So these are not rate limiting steps in this example. Rate of the enzyme 1 is the rate limiting step. So the conversion of A to B is the rate limiting step here, or sorry, A to B. And that is going to be determined by the amount of the product D. So if we have a high concentration of product D, that is going to turn off the action of enzyme 1. And if we have lower concentrations of product D, that is going to encourage the activity of enzyme 1. So in this instance, as we said earlier, A to B is the rate limiting step. Lastly, we have feed-forward activation. This is a much more rare occurrence. Feedback inhibition, just like with negative feedback, is much more commonly found throughout the body. Feed-forward activation is very rare. And again, just like with the negative and positive feedback, feed-forward activation is usually only a part of a larger negative feedback inhibition. So if we look at the feed-forward activation here, the actions of enzyme E1, so enzyme 1, which is catalyzing substrate A to product B, the levels of product B, if there's excess levels, that can actually feed forward and activate or sort of amplify the action of enzyme 3, which is going to help to clear the substrate C to turn that into product D. Okay, let me say that again. So if we have excess quantities of substrate B, that is telling us that there's a buildup happening in an earlier stage or step in the reaction. So in order to clear that substrate, we can feed forward and tell an enzyme that is upstream in the process or upstream in the reaction to increase its rate of catalytic activity so that it can clear the substrate C to D, and therefore enzyme 2 is going to further catalyze and eliminate the excess B and catalyze that into C. So feed-forward activation is another example of how enzymatic activity can be